Hi everyone, my name is Michelle Soupir. I'm a professor in Ag and Biosystems Engineering at Iowa State University, and I'm here today with Dr. Billy Beck and a PhD student in our department, Clarisse Siababa. And we are going to talk to you about one of our new projects funded by the USDA on wood chip bioreactors. So I'll start out by giving you a little bit of history. If you come to the state of Iowa 200 years ago, you would see a prairie and wetland system. But if you come today, you'll see a system that's highly agriculturally productive. And so the difference is that tile drainage has been installed. Tile drainage is very important. It's used to manage the moisture content in the soil, to allow crop production, allows more farmable acres, and also allows heavy machinery to be able to gain access to the ag land early in the spring when crops would be planted. So this is very important, but it's also created a situation where water is able to move through the system, collect nitrate that may be in the soil due to the high nitrogen content of soils or due to nitrogen fertilizer or manures that have been applied to the surface to fertilize crops. And then from there, that nitrogen moves into the tile lines and can move off-site rather quickly. So one strategy to try and improve water quality and to treat that nitrate-laden waters is a wood chip bioreactor. A bioreactor is an edge-of-field conservation practice that can be installed between the agricultural land and the stream, and it's used to intercept that tile drainage and treat it before it's discharged. If you go out to the field and you want to look at a bioreactor, there's actually not very much to see. You might see a control structure to help manage the water flow, but everything that's happening is actually below the ground. When a bioreactor is installed, a pit is dug and it's essentially filled up with a carbon source. Wood chips are the most common source that's used in Iowa and much of the upper Midwest. And then a layer of soil is placed on top, and then it's going to be planted to some kind of prairie or vegetation. So the bioreactor, the water routes through the bioreactor. It can bypass the bioreactor during periods of heavy flow, and that's to ensure that water isn't backing up to the crop field and potentially damaging the crops. And then it discharges to the surface water. Most bioreactors are able to treat between 15 and 60%, meaning that is the percent of nitrogen that is converted from nitrate, which is the harmful form that can cause a risk to public health, can contribute to downstream algae growth, and then remove it and convert it to nitrogen gas. The process that is taking place inside the bioreactor is what we're really interested in studying. So the bioreactor contains that carbon source, which is covered with microorganisms. It's actually the energy source for those microbes. And those microbes then work to take that nitrate and convert it to a harmless nitrogen gas. And that process is called microbial denitrification. As you can see, I'm not standing next to a bioreactor today. One really cool part about this project is that we get to think about the different trees and experience how we can manage our woodlands in order to provide the wood resources and carbon sources for the wood chip bioreactors. So today we're going to tell you a little bit more about our project, which is looking at those different carbon sources and specifically focusing on different types of wood species to try and better understand what are the extractives that are present in different wood species and how do they contribute to the microbiome or the community of microorganisms that all contribute to that denitrification process. An important aspect of this is, where does that wood come from? And so that is why Dr. Billy Beck is here, and he's going to talk to you a little bit about woodland management and how we can both manage our woodlands in a productive way and then use extra wood sources and wood chip bioreactors for combined ecological benefits.
Hey, I'm Billy Beck, Extension Forestry Specialist with Iowa State University Extension Outreach. And like Michelle said, one of the really neat parts of this project is connecting the water quality edge-of-field practice bioreactor facet with woodland management. We have over 3 million acres of woodlands in the state. It's a precious resource. Of those 3 million acres, 85% are privately owned. And that's important because we know from surveys and workshops that most of those acres under private ownership are passively managed. So Michelle talked a little bit about woodland management. So those acres are mostly passively managed. And in fact, we really need more of our acres to be actively managed. And I'll talk about what that means in a second. So forests, the 3 million acres of woodlands in Iowa, provide critical ecosystem goods and services to all Iowans. So things like clean water, clean air, wildlife habitat, and more importantly, the past few years, mental health benefits. And if we're not actively managing our woodlands are just not giving us the maximum that they can give as far as ecosystem goods and services. What we want to look for in this project is how do we sustainably source this material for carbon media for these bioreactors, but at the same time enhance woodland health by encouraging active woodland management across the state. And if you talk about forestry around the country and the world, markets tend to really help drive management activity. So one of the neat facets of this project is we're looking to see which woods or which blends of wood chips work best in bioreactors, and then how do we create markets for those species if they're not already a high dollar wood species in the state. So a lot of people think or ask, what is active woodland management? So in the past, 200-300 years ago, our woodlands were managed by a lot of natural and anthropogenic factors that we just don't have today. We've taken a lot of things out of the equation, things like fire and things like grazing from large mammals like bison and elk. And we've added factors such as invasive species, parcelization, other things that really we can't really do a hands-off approach anymore. We need to be involved in our woodland stewardship and our management for these woodlands to be resilient and for these woodlands to give us all the ecosystem goods and services that they provide. And when I say active forest management, it's really a catch-all term. And it really is based on what you do in your woodlands management-wise is really based on the landowner or the steward's goal. There's no one right way to do it. So active management generally entails things like controlling invasive woodland species, planting new trees, reforestation, thinning our woodlands so that more sunlight gets to the ground, harvesting our woodlands for sanitation, for economic return, or to achieve a stewardship goal like wildlife habitat or water quality. So again, the neat thing about this is how can we connect bioreactors to woodland stewardship using that residual material that we harvest, perhaps getting a higher market value for it, so that would hopefully encourage more landowners to perform active woodland management across the state. In past extension events, we talked about the physical characteristics of the chips and how they vary by species and how that could potentially impact bioreactor performance. The main goal for today is to talk about the chemical differences between the species and how that might impact bioreactor performance. So we're going to explore some cross-sections of trees and talk about how chemical characteristics may vary based on the species, based on the size or age of the tree, and based on the growth rate of the tree. And these are all species that are involved in this research project. So this first one is probably Iowa's most famous species. This is black walnut. And I want you to notice two different things here on this black walnut. This interior dark area is what we call heartwood of a log. This outer ring, which has a light color here, is what we call sapwood of the log. Both are very important for the overall health, growth, maintenance of the tree. So heartwood is important because, for this project especially, is heartwood, one of its main functions is that it houses byproducts of metabolism, which we collectively call extractives. So extractives, like you'll hear from Clarisse in a bit here, are kind of a catch-all term for things like fats, resins, terpenes, oils, and each tree species has a unique extractive cocktail mix housed within the heartwood. And it really is that mix is what gives certain species like black walnut its coveted color. It gives species like eastern red cedar that great smell. It helps with rot resistance. So a lot of cool things are derived from those extractives that are housed in that heartwood. We're interested in, if we chip this log up for these different species, how do those chemicals housed in the heartwood impact the microbiome of a bioreactor, thus impacting things like nitrate removal. So I want to point out three things here that kind of say that a tree is not a tree is not a tree, a log is not a log is not a log. The amount of heartwood, not only does the chemistry vary between species, but the amount of heartwood versus sapwood varies based on a number of factors between these species. So things like the age of the tree, the size of the tree, the growth rate of the tree, where we harvest the tree from, and how we process that material like chipping or tub grinder is going to impact how much heartwood versus sapwood we get in our bioreactor mix. And sapwood, very, very important for tree growth, but it doesn't house the extractives that may really impact the microbiome of a bioreactor. Sapwood is, as you can guess it, the living conductive tissue of a tree. So this is what brings the sap, which carries nutrients and sugar, up and down the tree for trees' overall health and keeping it alive. The heartwood, again, houses extractives, provides rot resistance, and is also for structural stability. In general, each species, when mature, has a set number of sapwood rings on the outside of the trunk. So that can vary. Some species, like this white mulberry here, only have a few sapwood rings on the cross section, where species like black walnut, green ash, and honey locust have maybe upwards of 12 to 13 sapwood rings on that outer part of the log. So species matters for the ratio of heartwood to sapwood. Let's talk about age, though. As the tree gets older, this is the base. This is about a 25-year-old black walnut. And you can see it's got its set number of sapwood rings on it and a nice big proportion of heartwood in the center. This is the exact same tree on an upper limb, so this wood is much younger than this wood here. And you can see the ratio of sapwood to heartwood is very different than the base of that tree. So if we were to include this material, say in a chipping of using an arborist chipper, where we use the log and the entire top of the tree, we would get a much higher content of sapwood in that mix than we would by just utilizing the log at the base. Also, the rate of growth matters. So this is a green ash. This is about the same age as this black walnut, but if you zoom in and look at the growth rings here, they're absolutely huge. That's going to impact the size of the heartwood in the center, the ratio of the heartwood in the center to the ratio of to the sapwood on the exterior. So growth rate really, really matters there. What's neat about these trees is that as the trees get older, the number of sapwood rings remain the same, but the heartwood material accumulates over time. So you're always going to have this area expanding and this area being, in general, the same number of rings. Larger trees are going to create more heartwood from their logs, provide more heartwood in their logs in general than younger trees. And that gets into the processing facet of this study. If we were to use an arborist chipper, like you see in municipalities, they're going to throw the entire tree potentially into that chipper. So you're going to get a mix of heartwood dense material, the base, but you're also going to get a mix of a lot of sapwood dense material, which is the upper limbs. Now, if we used a tub grinder, which basically you throw everything in, they're kind of like an arborist chipper, but they can take on huge diameter material, you're going to get much older logs potentially being able to be ground up, and you're going to have, again, you'll have tops of the trees and all that kind of stuff that's got a sapwood component, but with those larger logs, you're going to have a high heartwood component to those as well. The way we process these trees, along with the age, the species, and the growth rate, really can impact the chemicals that we're providing to the microbiome in our bioreactors.
Hey everyone, I'm Clarisse Siababa, a Fulbright scholar from the Philippines, and it's a beautiful day to be out in the field because, I'm usually in the laboratory. But yeah, I'm very grateful to be part of Billy and Michelle's project on the wood chip bioreactor. And now, as Billy mentioned, the wood properties and extractives are very important because they can affect the denitrification performance of single wood species. Thus, it is vital to understand the impacts of wood extractives and how they can affect the nitrogen and nitrate removal of each wood species. For this project, we have identified 12 wood species that can be used as wood chip blends that would be used for our pilot scale bioreactor. And for the wood extractives, they are very vital because they are the non-structural compounds such as the phenols, terpenes, fats, fatty acids that can affect the denitrification potential of our bioreactors. So as Billy mentioned, the heartwood and the sapwood ratio can affect the denitrification potential, especially how many carbohydrates, free sugars are available in our wood species, and how can this affect the microbiome of each wood species. In the laboratory, we are using the wood species that have been identified based on their abundance, their vessel distribution, and their durability. The wood species that we have are undergoing cold water solubility extraction that you'll be seeing later on. For the cold water solubility, this is vital in the process of understanding the wood extractives, and they can be divided into 3 different compounds. The first one are the polyphenols or the phenolics, which comprised of the tannins, the gallic acids, and the ellagic acids, which can somehow interfere with the bioreactor performance. And that's one of the objectives of the project. We wanted to know if the tannins can really interfere with the denitrification potential of the wood chips. The second one are the free carbohydrates such as the glucose, galactose, lactose, maltose, and cellulose, to name a few. And as mentioned by Billy, these free carbohydrates can be found in the sapwood instead of the heartwood, because basically the ones that we can find in the sapwood are the carbohydrates that is used by the woods for their metabolism, while the ones that we can find in the heartwood are the ones that have been used and as products of secondary metabolism, called the secondary metabolites. And the third component of our wood extraction are the secondary metabolites, which includes the syringic acid, vanillic acids, and other polyphenols that can also interfere. It can either increase or decrease the denitrification potential of our wood species. So overall, we wanted to know how these wood extractives can affect the denitrification potential of the wood species that we have identified. Our project aims to understand the impacts of wood extractives on bioreactor performance by characterizing single wood species. 3 cores from individual wood species were collected using an increment borer. Lubricate the borer by applying beeswax to reduce friction and insert the bead at breast height. Then turn it clockwise to penetrate the tree to the center. It then produces a pencil-sized core spanning bark to middle of the tree. The annual growth rings can be used to estimate the tree's age. The wood properties and extractives may affect the species' denitrification performance. These 3 cores will be used to analyze some wood properties and extractives. The wood extractives were prepared using cold water solubility extraction. The cold water procedure removes part of extraneous components such as inorganic compounds, tannins, and sugars present in the wood. Approximately 2 grams of oven-dried grounded wood samples were weighed and transferred to a 400 ml beaker, followed by the addition of 200 ml deionized water. Then, water is slowly added to the ground wood samples in the beaker. Extraction is carried out at 23 degrees Celsius or room temperature with constant stirring using a magnetic tabletop stirrer for 48 hours. While the samples were being mixed continuously, a vacuum pump setup was prepared with filtered crucible in a 1000 ml filter flask with a rubber flange and a vacuum source. The vacuum is then turned on and the wood mixture with cold water was slowly transferred to the vacuum pump setup with addition of the ionized water to transfer all samples from the beaker to the filtered crucible. The setup was allowed to stand until all liquid filtrate was separated from the solid particles in the crucible. The wood extracts were collected from the filtrate and they are stored in the glass amber bottles. A portion of the extracts were aliquoted in amber glass HPLC autosampler vials, for submission to the Iowa State University Metabolomics Research Laboratory for GCMS and LCMS of free carbohydrates and HPLC analysis for tannins. The remaining wood extracts were analyzed for colorimetric total phenolic analysis and nitrate analysis at the Water Quality Research Laboratory.
When we first started working on wood chip bioreactors more than 15 years ago, we really just thought if you dig a pit, get some wood chips in there, as long as the water can flow through the bioreactor, we should be in good shape. But we're seeing so many differences as far as how different bioreactors perform, and we believe it really comes down to what is the wood product or that carbon source that's in the bioreactor. You can see so many different things can come into account if you think about different species of trees, different ratios of heartwood and sapwood, how the tree was processed either at a mill or was the entire tree chopped up and then used as the carbon source. There's so many different things. And then the color of the tree and those different extractives that can come out. So thanks for tuning in today, and we look forward to keeping you posted as we learn more about the different things, the different extractives that can come out of the wood, and then how they impact the microbial community that's actually the workhorse, that's actually doing the work, converting the nitrate to nitrogen gas.
All right. Well, yes, please feel free to submit any questions you have for our folks today. So I'll invite them to turn on their cameras and join us for the discussion. There have been a couple questions already, so I'll toss this one out to maybe Clarisse, but is this study looking at one species at a time, or are you also looking at mixtures of these different species to see if you can optimize that denitrification?
So currently we are working with individual wood species, but we wanted to identify which wood species will have the highest and the lowest nitrate potential. And we're going to create a wood chip blend by mixing different wood species and potentially create basically a wood species recipe that will be best for our bioreactor.
And I know, Billy, you were talking about, I think when it was the chippers were up there, but as they're entering those chippers currently, is the bark being removed to just use the wood, or is it the whole thing going in and then you get both?
You get both. Yeah, debarking is an extra added step that could add cost. So all of it is getting thrown in whole. And I misspoke on that image there. That giant chipper you all saw was what we're calling an industrial scale chipper. It's not something you would see in a municipality, but it's not as big as a tub grinder, which is basically like a giant garbage disposal, often used after, disaster cleanup like the derecho and hurricanes, where like everything can get thrown in. So the arborist and the that larger industrial chipper are the kind of the two. Small, medium, and the large would be the tub grinder material, which is almost a shredded chip. But yes,
Well,
bark's included.
I'll toss this one out there between Clarisse or Billy, but if the heartwood holds rot-resistant properties, would including it into a bioreactor limit the microbial activity and therefore denitrification? And if so, is it feasible to separate the heartwood from the sapwood to then facilitate optimal bioreactor performance?
Right now, based on the current metabolites analysis that we have, there are actually rot-resistant enzymes that are detected. And based on the initial analysis that I did, these are the compounds that can interfere with nitrate removal, specifically for the four wood species that we initially used for our first batch. But I'm not sure how it works by separating the sapwood with the heartwood. Maybe Billy can help us answer that part. But definitely, we have identified some inhibitory compounds present on the wood chips.
Yeah, that's what's so exciting about this project is yes, they have rot-resistant capabilities, but we're going to find out how do those specifically impact the denitrifiers in the bioreactors. So
separating heartwood from sapwood would be very tricky and another added cost step. The one thing that might be a potential there is if you look at mills, so what they're milling up the log of the tree or the main stem of the tree, those are round tree trunks. So what they do is they'll cut the slabs off the side, and those really are limited, except for firewood, in their use and marketability. So those slabs, if you look at the cross sections that I show, would be primarily sapwood. So if we come to find out that X species extractives inhibit denitrification, maybe there's still potential with the sapwood component from mill residue.
Right. And I know, Michelle, this one had come up at our recent workshop, but there's a couple questions in here about thoughts of incorporating biochar as a mechanism within the bioreactor system.
Mhm. Yeah, so those of you who may not be familiar, biochar is produced through a kind of a high temperature process and can be based off of different types of grasses, different types of woods. So a lot of different types of vegetation can be used to produce biochar, even corn cobs or corn stover. And so we did do some experiments probably about 8 years ago. We had graduate student Emily Martin who worked on this, and we produced a number of different biochars from both different types of vegetation and also processed at different temperatures. And unfortunately, we didn't see an increase in nitrate removal from really any of the treatments when compared to just kind of the standard wood chip. I think that we just weren't able to see that kind of development of the community for denitrification and conversion of nitrate to nitrogen gas. There was a little bit of potential for phosphorus removal, so kind of the ability to capture the positively charged phosphorus may be an option for some of these biochars. Another challenge with the biochar is the size. Most biochar is pretty similar to the type of product that is first put through that pyrolysis process, and so it's very fine particles. And so we have concerns that that would lead to clogging in if we were to try and implement into a field scale bioreactor. There are some post-processing options, so you could potentially coagulate that biochar, but it would add cost. And so, to be honest, the literature, the scientific literature on biochar and bioreactors has been somewhat mixed. And so just from the cost perspective, it's not something that we've really been pushing or exploring.
There's a couple questions here, Billy, I'm going to toss your way. So there's a question about using deadwood like ash and then potentially using woody invasive species. So if you want to tackle those two topics for us.
Yeah, real quick, ash is a challenge, and that's a question we always get is why don't we just use the ash? Emerald ash borer has been confirmed, I believe, in all 99 Iowa counties, spreading east to west. So why don't we just use all the ash? So a few things which make it challenging is ash degrades fairly rapidly when standing in the field. So if we were going to use ash as a log, we would need to cut it and use it very either before it dies or very soon after. And that wave has passed. So many of our ash are very brittle. Loggers are hesitant to cut it. So that's the challenge with kind of rural ash. And I guess unfortunately, a lot of our historic city or urban plantings were almost monocultural in nature. So either maple or ash. We have a ton of dying ash in our cities. However, those tend to be more open grown. So I would say less easy to process and fit through a chipper. So if we did want to use urban ash, I think tub grinders might be the way to go. That's the giant garbage disposal that you can simply dump odd shaped large chunks of tree into. So that's kind of my thoughts on using the ash. It's going to have to be maybe a tub grinder situation. And as the tree breaks down, we really don't have a good grasp on how that impacts extractives, which is what Clarisse is diving into. So, as far as invasive species, the list of species that we're looking into with Clarisse's facet of this, one of them is white mulberry, which is a non-native mulberry. We actually have a native mulberry in Iowa, but a non-native mulberry that is a tree form that does have a volume we can fairly easily cut, transport, and use. Other invasives like bush honeysuckle, non-native buckthorn, roundleaf bittersweet, which is a vine, they're often smaller diameter, multi-stemmed. So obtaining the sheer volume needed to fill a bioreactor of those would be very, very challenging. But like Clarisse said, if we do investigate those and there is some potential for enhancing denitrification, maybe we add 5% to the mix or 10% to the mix. So it doesn't have to be, 100% honeysuckle. Heard a great idea on Tuesday during a workshop is we put a bag on our forestry mower heads, these big machines that grind up invasive species in the field like a lawnmower, and we capture the honeysuckle as it comes out the back. So if that person patents that, that's their idea, not mine. But the size of the growth form of the invasives make it challenging.
All right, there's a couple questions here, Michelle, I'm going to toss your way. So what is the typical size and volume of wood chips needed, and how leaves may impact a bioreactor function?
Yeah, so if we go back to kind of the design standard that's set by NRCS, we want the a diameter of a wood chip to be at least 1 inch in one direction. And so that's kind of the criteria when installing a field bioreactor and making sure that the wood chips are of the appropriate size. We would go back to currently that design standard. As far as how big is a bioreactor, one of our students, Alex Buseman, is actually on the call today, often compares it to like a school bus. So a buried school bus. When the bioreactors are installed, often we would have more than one semi truck full of wood chips to fill a bioreactor. Not uncommon to have two. If we're working with a larger drainage, maybe 100 acres or larger, sometimes we'll even design a dual chamber bioreactor. So that would be two bioreactor pits that are right next to each other. And each of those may need, more than a semi truckload full of material. So it is a large volume, and that's why we really want to see the kind of the workforce development and more suppliers of this wood material, so we're not trucking the wood chips from so far away, sometimes over 100 miles to transport wood chips from an appropriate facility to the site of a bioreactor installation. As far as leaves go, that's kind of leaves and other kind of small particles. We tend to try and keep that out of the bioreactors. The main reason being that as those break down, we don't want them to clog the bioreactor. So it's really important that water can flow freely through the bioreactor. If you think about a tile drain and how fast that water is moving, especially in early spring, when fields are kind of dewatering, uh, we don't want the bioreactor to cause any constriction or to slow down that flow. We want the water to be able to move freely off the field so it doesn't back up into the cropland. And also just so that we can treat the highest volume of water possible. We've really found that the more water that can move through the system, the better, because we're able to remove more of that nitrate load. So um, we do have a project funded by the Iowa Nutrient Research Center where we are looking at some of these other types of grinding strategies, kind of more like shredded wood, or we have also learned actually through this project that the wood chips are screened. So they're chopped up and then they're put through a screen in order to make sure that that minimum 1 inch size that NRCS standard is met before those wood chips are sent off to be installed in a wood chip bioreactor. And so one of our other projects is looking at unscreened wood chips. So a combination of both those bigger sizes and smaller sizes. And these projects are looking at both the nitrogen removal as well as using some tracer studies to kind of look at the flow through these bioreactors as they age in hopes that we can eventually kind of open up the options for different types of grinders or wood processing facilities to be able to provide wood chips for bioreactors.
Okay. I'm going to toss this next question towards Billy. So as you're examining the different species of wood and maybe a recipe of chips, as Clarisse mentioned, how do you see this being implemented on a large scale? Do you see potentially using only like localized species, especially in areas where there may not be a wide variety of species?
Yeah, great question. One of the most exciting pieces of this study is the supply chain. Um, so I would say that, we're looking at species, but we're also looking at like groups based on physiology. So, some species have their vascular system. We can break a lot of our Iowa species into three ways that, for example, their vascular system is arranged in the organism itself. So there's, like Clarisse might have mentioned, there's ring porous. And I actually have a bonus surprise slide if you want to show what that is, Liz, if we have time. But diffuse porous and semi-ring porous. So in our initial lab test, many years ago, um, there was definitely a difference in species as far as nitrate removal, but we couldn't figure out why. We knew there was a difference. We couldn't figure out why. And it wasn't based on density like our initial hypothesis was. The only thing the two higher performing species had in common was that they were ring porous species. So I think maybe looking at the physiology groups, perhaps not the species, or if the species have similar extractives, the nature of their extractives is the same. Um, so that's how I think we're going to handle the geographic spread across Iowa.
All right, Clarisse, I'm going to direct this one to you. So this is an anaerobic process. There was a question about that. So lack of oxygen. But as we're looking at these different species, are we aiming to get an optimal denitrification rate, or are we also looking at like longevity? And feel free for others to chime in as you're kind of exploring these different species.
Okay. Basically, ideally, we want to have like a complete denitrification process because it means that there's reduced, greenhouse gas emission as well. But we also want to make sure that there is longevity in the process, that this wood chips that we're going to use as carbon source can be, um, long standing there in like for more years to come, like not just for 1 year or 2 years, because technically that's the reason behind using bioreactors as edge of field technology. But aside from microbial denitrification, we wanted to look into what are the other, biochemical pathways occurring in the system, because you've mentioned that it is pretty anaerobic there, but we want to know if the nitrate is totally reduced to nitrogen, or does it produce ammonia, or does it produce other gaseous, components that leads to incomplete denitrification.
All right, so Michelle, there's been a couple questions here related to this in terms of the lifespan of bioreactors and what do you do with the chips at the end of their lifespan?
Yeah, absolutely. So, our estimates of the lifespan are 10 to 15 years. Some of our earliest bioreactors were installed kind of in the 2010 timeline, maybe a few earlier than that, but we're just now getting to kind of that threshold for many of the early bioreactors that were installed in the state of Iowa. And it's been really interesting. We've seen some sites where the wood chips seem to degrade rather quickly. So maybe at the shorter end of that period, some ways that we can tell that that's happening is we'll see slumping of the bioreactor. So if you go out to where the pit is in between kind of those two control structures, you'll see that the ground is really kind of sunken in, and that's something that's an indication that the wood has been breaking down and that we're probably not getting good flow. I talked a few minutes ago about kind of the importance of having good flow through that bioreactor. And as that wood material is breaking down, water probably can't move through. In one particular site, it was almost like soil or compost. And that site had not been installed that long ago. But there are other sites that some of our students have gone back and revisited many years later, even on the upper end of it, 15 years, and they're still functioning very well. Water is still flowing through the system, and we're still seeing good denitrification. And even in some cases where we've had some clogging of the bioreactors, if we take the wood chips and bring them into the lab, we still see that nitrate removal can occur. The microbial communities still seem to be active and seem to have enough carbon. So that longevity question it's a really tricky. Something that we're really hoping to lean into a little bit more and better understand that if we can choose the wood species to at least have some species in there that have a longer lifespan and can kind of maintain that wood structure, that would be ideal. Understanding kind of the water chemistry and what might be leading to the breakdown of the wood chips that exist. And then also just kind of getting a sense for what kind of indicators could we use to know when a recharge might be necessary. Maybe we don't see the slumping, but if the microbial community is no longer functioning, that could be another way to try and understand if we need to go ahead and recharge that bioreactor.
There's another question here while I got you on kind of design and flow. Is there a max flow rate or minimum residence time that's kind of your target for keeping this bioreactor functioning at the level you want it to?
Yeah, it's some earlier work that we did kind of targeted an 8-hour residence time. So a drop of water would be in the bioreactor for about 8 hours. And that was a sweet spot because we were looking at kind of optimizing the nitrate removal, but we also wanted to minimize the potential for greenhouse gas production, both methane, which can be produced if the bioreactor is flowing too slowly. So if the water's in the bioreactor too long, as well as nitrous oxide, which is a byproduct of incomplete denitrification. So if the water is rushing through too quickly. So 8 hours is kind of a target. NRCS is looking at, 15% of peak flow. And another important point, though, is that, as I said earlier, the more water that moves through the bioreactor, the more nitrate mass is removed. And so at the end of the day, we're really trying to remove as much nitrogen as possible. And we want these conservation practices to be put to their full use and full potential. And so by being able to make sure that the water can flow freely through the bioreactors and just trying to maximize the amount of water that can flow through, I think is a really good strategy as well.
So, Billy, kind of going back to those chippers and kind of right now, all these chips are being trucked in. There's a question here, kind of just how do you get these wood chips to the site? So right now they're being trucked, but would there be potential if we can open up some of these other grinding options to maybe even do that on site?
Yeah, I think there is. Again, like 2 semis it takes of chips to fill a standard reactor. Um, but if you look at like, a 5 acre woodland and some of the material that can be thinned out of that woodland, those smaller 5-10 acre chunks that are often on farms, um, that could be a source. It would just come down to getting the equipment there, having that network local to either like maybe a co-op where farmers could share the equipment or something like that, to get that equipment on site and then the time and labor to get it in there.
And for clarification, you're not talking about clear cutting that 5 acre area.
No. And actually, I had some bonus slides that I can maybe share in a blog later, but we're not suggesting at all going out and denuding the landscape of trees. And even if we do identify, say, 3 trees, we're not going out and, targeting those species for complete removal. Our woodlands in general in Iowa are growing about 2.5 times more volume per year net versus what's being removed via harvest and mortality. So the wood is out there, and a lot of the woodlands are increasing in small diameter material, often of trees that grow in the shade, grow well in the shade, like a lot of the species we're looking at in this project: hackberry, basswood, sugar maple, that have low marketability as a species and even lower marketability as a size. So by removing those as part of active forest management, you can get more sunlight to the ground where more, I'll say, quote, desirable species, often in the landowner's mind, like oak, and walnut, the seedlings can use that sunlight to regenerate. So, and nothing against those species that we're looking at removing, but we're losing our oak woodlands to the tune of about 5,800 acres a year. So active forest management is needed to get that oak back on the landscape, and thinning our woodlands and harvesting our woodlands is often a critical piece to get enough sunlight for those young oak seedlings. So it's all about sustainably sourcing these things. We've got the practices, we've got the volume. It's just about getting them to the bioreactor site.
Awesome. And there was a question here about potentially using bioreactors in an urban setting for different runoff and things. And I'm actually going to share a link here to a webinar that Laura Christensen had done where she looked at a whole bunch of different types of webinars, or sorry, bioreactors for different removal processes. And so I encourage folks to check that out, as most of our bioreactors here are going to be for tile drainage, infiltrated water versus runoff water. And if anyone would like to make a comment onto that, feel free. Okay. So Billy, while I got you here, there's a question here. How many mature trees would it take to fill a semi-load of wood chips?
Yeah, great question. We actually did some tests. I see Troy Heron is on here. He was very instrumental in doing these tests where we took a known volume log, threw it, put it in a chipper, and then measured the output. That's going to vary by species. Shocking. It depends, like we said. Some species chip up very large because of their density and growth characteristics, like shagbark hickory, for example, produces a number of large chunks. So its chip distribution is swayed to the larger side. Softer species like cottonwood produce a more uniform, smaller chip. So species is gonna matter. But our quick back of the envelope calculations, a standard bioreactor is about 100, maybe on the smaller side, 100 cubic meters or 100 cubic meters of material. That's about 69 to 100 logs that are about 16 foot in length and 13 inches in diameter. So that's kind of the 10 to 13, 14-inch diameter stuff is kind of what we're looking at. So it does vary, but that's kind of a general ballpark. But the U.S. Forest Service has data around that is collecting data every year in Iowa, and they report every 5 years, forest inventory and analysis. And you can look up the estimated species and size distribution that's growing in our woodlands. And we use that to answer that question, do we even have enough of these species to fuel the bioreactors needed? So,
Because we're talking on the scale of 100,000 plus bioreactors needed to meet some of our statewide goals here.
right.
All right, we've got a few more questions, but I want to share my screen one more time. For those of you that joined us today for CCA CEU credits, feel free to email me. My email is there on the screen, and I'll put it in the chat here real momentarily. But that's your name, your CCA number, and if you use a different name to log into today's virtual field day, please be sure to share that with me as well so we can get that credit submitted on your behalf. Please do, though, email me by 5 p.m. today, and I'll get your credits submitted for you. And if you're a regular on our online offerings, you've seen this slide before, but we are asking folks to voluntarily complete a demographic survey. You can do so by either scanning that QR code on the screen or visiting iowalearningfarms.org/survey. If you've already done it for a previous virtual field day or webinar, thank you. You only need to do one a calendar year. So appreciate all of you who have already completed that so far this year. And my last slide here is to invite you to join us on March 12th. We've got Dr. Dan Anderson, Dr. Raj Raman, and Philip Rockford to talk about manure application and cover crop planting timing to maximize water quality benefits in corn. So switching gears a little bit to talk more water quality from an in-field management perspective. So let's see. One of the questions was, and I'll toss this out to the group, whoever wants to jump in first. When do you think you will have information on the best mix for the bioreactor?
Right now, maybe I can answer this. So right now we have initial candidates, but we want to make sure that we run all tests first before we identify those top candidates because we are going to use them for the blend that we were talking about and further use them for column bioreactor first before the field bioreactor setup. So right now we're working on the analysis of the individual wood species on benchtop setup, like mason jar setup first, and then upscaling it to column bioreactor before out in the field. So watch out for those guys. We are going to update you on the best wood species to use.
So we'll be inviting folks back probably either as a virtual field day or a webinar. So you can join us for those. Those are every Wednesday at noon. And I'll maybe wrap with this as a last question here because I had to search it up and I'm not sure I'm going to pronounce it right. But essentially, could this research on your extractives and how things interact be used to impact the understanding of the hugelkultur techniques, which is like the mounded gardening method with layered woods and things? Are any of you familiar with that? Okay, so maybe if someone wants to run with some of the data that gets used here and how that might influence that particular practice, maybe that's an added bonus to this research project. So if there are no other questions, which I'm not seeing any at the moment, I want to thank you all for joining us today, and thank you to Michelle, Billy, and Clarisse for your time. Please keep in mind that you'll be getting a recording of this afternoon and can share it out with folks, and we'll see you next time.